Dr. John Bennett from sunny Miami, Florida. Tonight we have the pleasure of having Shahriar Yaghoubi, PhD, MBA from UCLA. He's going to talk to us about what he's done and specifically about Cell Site Technology, a company that he's working for. Uh, and after the presentation, we'll open it up for questions if anybody has any questions. And this is on medgadgettv.com live. Good evening, Shah.
Good evening, John. Thanks for having me on this interview. As you mentioned, I am Shahriar Yaghoubi. I'm the Chief Scientific Officer of Cell Site Technologies. Cell Site Technologies is a molecular imaging company that started operations in 2010. It was a spun out of Dr. Gambhir's laboratory at Stanford. And currently, our laboratory is located at China Basin on the UCSF campus. Mm-hmm. And we are also part of the QB3 organization.
When it first started, Shah, was it specifically for a reason? Was it, or well, I guess I understand that you guys were using reporter gene tech. Is that what started the company? Was there one specific thing that made it start?
Yeah, so I had been working with Dr. Gambhir both at UCLA and Stanford, developing imaging tools for monitoring cell kinetics.
Okay.
And by cell kinetics, I mean non-invasive imaging tools for tracking where the cells migrate, their trafficking, tracking cell proliferation, tracking cell survival, also quantification of cells at a certain location, as well as figuring out cell differentiation and cell function. So these are the technologies we have been working on, and reporter genes was a main technology that I had worked on different models in both autoimmune diseases and oncology. So the company started by bringing in reporter gene technologies to help investigators monitor cell kinetics.
Okay, I'll just put up the website and you can take us through it. Sure. Let me see here. Okay, let me start with the first page, and you just direct me where you want to go. Okay.
Yeah, so if you want, you can go to About Us. I give a little bit of a history.
Where do you want? About Us?
About Us, yeah.
Okay. Okay,
so Cell Site, the company was founded by Sam Gambhir, myself, and Sam Quesada. And then it started operating when Aruna Gambhir joined the company as the CEO in late 2009, early 2010. I think a lot of people are familiar with Dr. Gambhir. He's a pioneer in the field of molecular imaging and has made a lot of advances in reporter gene technologies as well as nanotechnology. And so we can go to the next slides.
Okay, which page do you want to go to?
We can go to the technology page.
Okay.
So as we mentioned, Cell Site is started by bringing reporter gene technologies, and we have done several projects with pharmaceutical companies using reporter gene technologies to track cells. And we have also done collaborative studies with UCLA, the laboratory of Dr. Chernin and Dr. Radu, developing non-immunogenic PET reporter gene systems, and also worked with Dr. Lawrence Cooper at MD Anderson and Dr. Amer Najjar, developing a reporter gene technology that was originally developed at Sam Gambhir's lab for clinical applications. And the one that I'm talking about is actually FHBG. FHBG is the first PET reporter probe to get an FDA IND to image cells in humans. And in 2005, 2006, we were able to demonstrate by collaborating with a group of Dr. Michael Jensen at City of Hope, we were able to demonstrate for the first time that a reporter gene can be used to image cells injected into a patient. And in this case, the image you see is the image of a patient with glioblastoma, stage 4. And this patient had recurrence and surgical resection. And after surgical resection, the T cells expressing the reporter gene herpes simplex virus 1 thymidine kinase had been injected into the site of resection. And then we imaged this patient with FHBG, and we were able to see a signal at the site of injection. And we were then able also to see a signal from another site. So the cells had actually migrated from the site of injection to a tumor in the corpus callosum. And FHBG had been able to detect the migration of these cells to this tumor. And this was confirmed by MRI at City of Hope that another tumor had been formed in this patient.
Mm-hmm.
So this was a very exciting study, and one of the main technologies that we originally brought to CellSite. And then since 2011, 2012, we have been funded by the National Cancer Institute through a SBIR grant to develop another PET tracer that was again developed at Stanford in Dr. Gambhir's lab by Mohammad Namavari. And this PET tracer is called fluorine 18 labeled ARAG or FRG. And currently we have a trade name for it called VISACT, standing for Visualizing Activated T Cells.
Mm-hmm.
So what they had shown at Stanford originally is that they had shown that if you treat mouse T cells with PMA ionomycin and then do an uptake assay with FRG, you could see significantly higher accumulation of FRG in the PMA ionomycin treated T cells, so activated T cells. We brought FRG into CellSite, and CellSite has an exclusive license on this tracer. And we have developed this tracer by testing its specificity, and we find that it has a specific accumulation in both activated human T cells and activated mouse T cells. We have also tested this tracer in a mouse model of anti-tumor immune response, and we've been able to detect signal in the tumor when there is an anti-tumor immune response. And the image you see right next to it is the image of the first image of FRG in a healthy human volunteer, showing what is the normal distribution of FRG in a healthy human volunteer. And you can see that there is distribution in the liver, the kidneys, the bladder, a little bit in the heart, a little bit in the salivary glands, and a little bit in the thyroid gland.
Okay.
But the main distribution is seen in the kidney. So we are now actually planning to start trials with FRG in cancer patients who will be undergoing immunotherapy.
Mm-hmm. Now, immunotherapy is a growing field, correct?
Yes, yes. There are now a lot of big pharmaceutical companies and small biotechnology companies involved in developing different types of immunotherapies, cell-based immunotherapies and antibody-based immunotherapies.
Mm-hmm. Okay.
And and one of the advantage of this tracer, uh, I think there are different advantages between the both technologies. So the reporter-gene technology is able to track basically every aspect of a cell that is injected into a patient, because it does require inserting the reporter gene into the cell. The advantage of FRG is that it doesn't require any kind of genetic engineering. We are basically imaging activated T cells that are present inside the body of a patient.
Mm-hmm. Very good. Okay. You want to stay on this page or move to another one?
I think we can move to maybe application.
Okay.
So, so we have been pursuing VISACT or FRG for the application of monitoring anti-tumor immune response after initiation of immunotherapy, and we have particularly um, investigated it in cancer immunotherapy, and that's our immediate goal. Um, but this is actually a very general probe which could have multiple applications. For example, it could have applications in diagnostic applications in autoimmune diseases. It can be used to detect, for example, um, inflammation in rheumatoid arthritis. And we have worked with UCSF, the group of Dr. Um, Ben Frank and Henry Van Brockman at UCSF have shown that you can image acute and chronic inflammation in the paws of mice with FRG. And uh, if we could basically pursue different type of applications with VISAC.
Could I ask a question, Shah? Is the primary way of determining response to immunotherapy blood work, like having blood work to determine if cells are going up or down or whatever?
So, so as far as I know, the the main way in the clinic used to determine response to immunotherapy is a modification of the RECIST criteria.
Okay.
Because it is known that immunotherapy behaves a little bit differently. So, but still, they do imaging studies and they see whether there is tumor regression or cancer progression.
Mm-hmm.
However, with immunotherapy, it could take a very long time to measure that response. It could start as early as 8 weeks after immunotherapy is initiated, or as late as 6 months, 8 months, or a year.
Okay.
So, and then when they were doing the studies in immunotherapy, they realized that the response could be very different. In most other therapies, they had noticed you either see regression of the tumor or you don't. But with immunotherapy, for a long time, you may actually see the tumor grow before it starts regressing. Or sometimes you will even see metastases, like formation of different tumors at other sites, before you actually see the tumor shrink. So right now the response is being evaluated by a modified version of RECIST criteria. Um, I'm not completely familiar with everything investigators do, immunotherapy investigators do, but I know they also do testing of the blood to see whether there are activated T cells in the blood. Um, I don't know if finding activated T cells in the blood has been very useful in figuring out whether there would be a response.
Right.
Um, the other thing is that in cellular immunotherapy, for example, they have noticed there's something called the cytokine storm. And I also don't know if that has been associated with a better response. It's definitely associated with a major side effect. So So we think that imaging could be useful for early detection of response, but these all have to be studies in clinical studies.
Okay. As the page says, I imagine the pharmaceutical companies are very interested in this.
Yes, we've talked to several pharmaceutical companies. I don't know if I can say their names,
Okay, you don't have to.
uh, but we have had major interest from different pharmaceutical companies, both in regard to cancer immunotherapy, but also in regard to some autoimmune diseases.
Okay, very good. Okay, you want to stay on that page or move somewhere
Uh,
else?
I think we can. I think that's probably it about the technology.
Okay. Well, you're partnering. Let's see, your partners in this project are a couple of big names there.
Yes. So we've had a grant with UCLA. It was an academic industry R1 grant. So we partnered with UCLA in developing non-immunogenic reporter gene system. We partnered with MD Anderson in taking reporter gene systems for clinical applications, but also testing VISACT or FRG in a CAR T cell therapy model. We we are a major partner with UCSF. All of our uh, clinical and preclinical studies are being done with UCSF, and we are also working with Stanford, and they they will probably soon start their own independent clinical trials with VISAC for immunotherapy monitoring.
Well, Shah, are all these institutions also involved in cancer immunotherapy? It's kind of like like we mentioned before, it's a growing area of study in science. But are a lot of places, are all those places involved too with cancer immunotherapy?
Yes, these are actually only some of the major institutions involved in cancer immunotherapy. I know at UCLA there is a major work being done on cancer immunotherapy. One of the lead investigators is Dr. Tony Ribas.
Hmm.
Uh, and then in MD Anderson, we were working with Dr. Lawrence Cooper. He was involved in cell-based immunotherapy. At UCSF, there are a lot of studies going on with checkpoint inhibitors and such as anti-CTLA4, anti-PD1, anti-PDL1, and also different types of antibody immunotherapies. And at Stanford, I also know there are different types of cell-based immunotherapies being pursued, as well as other types of immunotherapies. Uh, but another interest actually comes from radiation oncologists because to some extent immune system is involved in the in in, in the radiation treatment for cancer, and and they are also combining radiation treatment with immunotherapy to get better results. So they are very interested in using VISAC. um, I know Dr. Gambir's app, although I don't know exactly what results they're getting, but I know they're working on graft versus host disease monitoring at Stanford. So their interest would be monitoring graft versus host disease with VISAC at Stanford.
Do you communicate with these other universities that are doing cancer immunotherapy, or you kind of stick to yourselves as far as what you're doing?
Uh, well, we like to collaborate with everybody.
Okay, so you communicate. How do you communicate with them? Emails, phone calls. How do you do that?
Well, I know there are major immunotherapy being done at Memorial Sloan Kettering. We haven't started any collaborations with them. It would be great to have collaborations with them. It would be great to have collaborations with Juno Therapeutics, for example, who is working with Memorial Sloan Kettering. And I know there is also immunotherapy going on at the Fred Hutch Cancer Center in Seattle.
Hmm.
Um, but there are also other centers, but we haven't started collaborating with the other groups yet.
Well, I was just going to suggest this platform is a great way to network with other scientists. I find it tremendously useful.
Yes.
But yeah. Now your background in molecular imaging, is that what really kind of brought you to all these like gene expression and cancer immunotherapy?
Yeah. So I went to UCLA to get my PhD in pharmacology.
Okay.
And, I originally started working with Dr. Chernin there, uh, and who was doing PET imaging studies in cardiovascular disease at the time.
Okay.
And I enjoyed it a lot. His laboratory was mainly focused on clinical investigation, and
But
uh,
the imaging part of investigation, right?
so yes, the imaging part of the investigation. And then
Okay.
I had several classes with Dr. Gambier in pharmacokinetics, and I was extremely impressed by how he he taught pharmacokinetics.
Okay.
And that's why I decided to join his lab, and I think I made a very good choice. And
Okay.
that's where I got involved in imaging gene expression. I started originally on developing imaging technologies for monitoring mRNA expression with antisense oligonucleotides, but I think that the technology was not advanced enough uh, to work for imaging, and as far as I know, progress has been very little on that field.
Okay.
So then I got into using reporter genes for imaging gene expression, and and I was very interested in clinical research. So I was very happy to have the chance to get an IND for FHBG and take it into clinical research. Uh, and then when I moved to Stanford, I started working on imaging technologies for monitoring cells.
Okay. Well, I'm a typical doctor. I don't know what the heck a reporter gene is. Can you briefly tell us what a reporter gene is?
So a reporter gene is basically a gene that you have some kind of probe that you can detect the product of that gene.
Yeah.
For example, a gene makes encodes an mRNA, and then that's translated into a protein. So if you have a probe that can detect either the mRNA or the protein, but mostly the protein, then you potentially have a reporter gene. Now,
Okay.
what you can do is that you can link the expression of this reporter gene to different types of molecular events that happen inside the cell. Like,
Okay.
for example, if you want to see if some promoter gets upregulated, you could put the reporter gene in front of the promoter, and because you can image the reporter gene, you can see whether that promoter gets upregulated or downregulated.
Oh, wow. You kind of determine function that way.
Yes.
Wow.
So you can use this for a variety of applications. You can put the reporter gene inside the cell, and because you can detect the reporter gene, you can at the same time detect the gene,
Oh,
uh, detect the cell. I mean.
that's fascinating. We'll have to have another show just for that. Well, okay. Let me check really quick. We're just starting this tech. I think most people don't know they can ask questions. Hold on. Let me see if I questions. Yeah, someone's asking a question, Shah, of where do you see the field of cancer immunotherapy going? Where do you see the future of cancer immunotherapy?
Um, can you hear me still?
Yeah, yeah, I can hear you fine.
Okay, so I'm kind of in the field because we develop tools for the field, but I think there's already been a lot of progress made in immunotherapy so far. Um, like for example, in melanoma and lung cancer, there are now immunotherapy drugs available. I know there are immunotherapy drugs available for prostate cancer, for neuroblastoma. There has been a lot of progress made, and based on the research that is going on in basic science, I think there's going to be a lot more progress made.
Okay, so the trials are encouraging.
I think the trials have been very encouraging with immunotherapy. Um, for example, they're getting very good responses with T-cell based immunotherapy in melanoma.
Mm-hmm.
So one would expect that would be coming soon
Okay,
for clinical application.
very good. Okay, very good, Shah. I really appreciate you taking the time to explain what cell site technology is doing, and we hope to have you on for more hangouts in the future.
Okay, great. Thank you for having me.
Thank you. Good night.
Good night.